I'm Andrew Huberman, and I'm a professor of neurobiology and ophthalmology at Stanford School of Medicine. Today, my guest is Dr. Noam Sobel. Dr. Noam Sobel is a professor of neurobiology in the Department of Brain Sciences at the Weizmann Institute of Science.
Information then synapses at the olfactory bulb from the olfactory epithelium. And the pattern of that synapsing follows what's referred to as the most extreme case of convergence in the mammalian nervous system. More specifically, what happens is that all the receptors of a given subtype, and remember in humans we said we have about 350, in the mouse we have about 1,200 probably. So all the receptors of one subtype converge to one location in the bulb. And this location is referred to as a glomerulus, or in plural, glomeruli. And that may be a slight oversimplification. It's in fact two glomeruli. There's a mirror, sort of a mirror cut line. And so all the receptors of one subtype will converge to two mirror glomeruli on the olfactory bulb. So you end up having two glomeruli that reflect that one receptor subtype. And so and this is as far as, I'm giving you now the textbook view of how the system works, but then I'll happily share with you things that pose a problem for the textbook view of how things work. But the textbook view of how things work is that every such receptor subtype is responsive to a small subset of different molecular shapes, what's sometimes referred to as odotopes, the molecular aspects of the odorant. So each receptor is responsive to a different subset of odotopes, let's say 10, and each odotope will activate a different subset of receptors. So potentially you have this insane combinatorics of this potentially 350-dimensional space in the human, potentially. But then because of this convergence, you end up having on the bulb, in a way, a map reflecting receptor identity. So let's say this coffee activates receptors of type 1, 3, and 7. So the glomeruli of receptors 1, 3, and 7 will light up, quote unquote, when I smell the coffee. And if you could take a snapshot of that, theoretically, you would have the map of coffee and so on and so forth. This is sort of the textbook view of how the system works. And then information goes from the bulb to several targets in the brain. I mean, what is referred to as primary olfactory cortex is piriform cortex and entorhinal cortex. This is on the ventral surface of the brain, the lower portion of our temporal lobe. And information goes there directly, but it also goes directly to the amygdala. It probably goes directly to the hypothalamus. It may go directly to the cerebellum. It goes all over the brain. So information projects widely from there. And as far as people understand, the map that may exist on the bulb doesn't exist in the rest of the brain. And the understanding of how coding occurs in the rest of the brain is murky.